Hello, my name is Anne-Marie Helmy. I worked on the col1a1 protein. This is a gene that codes for the alpha-1 chain of type 1 collagen, which is very important for the structure and strength of cells in our body. Col1a1 is found in the extracellular matrices and endoplasmic reticulum of our cells. Because this gene is critical for bone formation, mutations can often be related to bone fragility. One mutation that we will discuss later on in this video occurs at the 836 position of the gene, and it is known as Caffey's disease and is characterized by excessive bone formation and ultimately bone fragility. So let's start talking about the shape of our protein, first with the primary structure. So here I just have a linear sequence. We can see that there is a little G at like every third amino acid. I included this because in my research I found that glycine is about 1/3 of the amino acids that make up the primary sequence of type 1 collagen. And these little white ribbons here are just representative of non-mutated R groups in this primary sequence. In reality, there would be one for every amino acid. This is a simplified version. Um, and so we would begin to see a secondary sequence as this primary sequence chain becomes begins to form shape and twists, and ultimately this alpha chain, as it twists and takes shape, will find two other alpha chains, one identical to itself, that is also coded by col1a1, and another chain that represented here in blue that is coded by col1a2. So again, those are two red chains coded by col1a1 and one blue chain coded by col1a2. Here's the R groups interacting with each other. They form this triple helix, and this is our tertiary structure. It's a very tight, strong structure. And as these triple helices begin to come together, we get a quaternary structure in the form of a fibril. Um, these fibrils include many triple helices. Um, yeah, so that covers the shape of our protein. Let's start to discuss our assigned mutation, which is Caffrey's disease. So as we saw here, this is a regular non-mutated primary sequence of our coded alpha 1 chain. And so what we see here is at the 836 position, there is a different R group. So in the non-mutated version, this R group is arginine. Arginine is a very basic amino acid, and it is involved in a lot of acid-base chemistry, which forms pretty strong bonds. It's also a pretty big side chain, um, but this gets replaced by a little cysteine here, and this cysteine differs from arginine in one, that it is smaller, but two, that it forms disulfide bonds when it interacts with other cysteine side chains. These disulfide bonds are very strong covalent bonds between sulfur atoms. And so what ends up happening is that you get a very, very tight alpha helix when this forms a tertiary structure. And this shortens the diameter of the alpha helix, which changes the shape of the protein and its overall function. And what happens is that there begins to be overactive col1a1 and overproduction of collagen, which ultimately makes the fibrils improperly structured, and this makes it unable to support the extracellular matrix, and the cells are unable to properly connect to other tissues. This can cause swelling and asymmetrical bone formation and bone bridges, pain, and fragile bones, particularly in infants. There's just another look at kind of the really tight diameter that that mutation makes and how that can really mess with the function of our cells. So some limitations. Our model is not perfect. The scaling is not accurate. It would, in reality, these are very small structures, microscopic. Um, Additionally, in reality, there are over 1,400 amino acids, much more than this small 15 amino acid sequence. There's much more variety of amino acids. I only really discussed glycine, arginine, and cysteine in this video, but many more amino acids take play here. And also, what really characterizes collagen is a very strong, tight alpha helix that is not really represented in this flimsy sort of model, but yeah, those are my limitations. Thank you so much for watching.